Great, everyone. Well, thank you and welcome to our webinar. We'll be talking with our colleagues from Privacy Analytics. My name is Rebecca Lee. I'm the Executive Director of Vivli. I'll be joined by Neve McGinnis from Privacy Analytics. She's a Senior Analyst from the Clinical Trial Transparency Unit of PA, and I'm really delighted to have all of you on today. We'll be beginning shortly. After the presentation, we'll be sending a link to the recording and slides. Just a reminder to post your questions via the chat, and we'll respond to your questions after the webinar. All of you will be muted during the time of the webinar, and we'll be taking questions from the chat. You can feel free to send questions during the course of the webinar, and we'll be answering them after the webinar. So thank you again for joining us today. Today, the topic of our webinar is really data sharing and anonymization, a hot topic, and we'll be talking a bit about our partnership, and then we'll be ending with, really what we're doing in this era of COVID-19, a challenging time for all of us during this pandemic, and in some small way, what we'll do during our partnership to what we're looking forward to doing in a small way to mitigate the challenges ahead. I'll be starting off and then handing off to Neve McGinnis. Thanks again. So our agenda today is really talking about the importance of data sharing and secondary use of data and the role of data sharing platforms in general. Then moving forward to talk a bit about the regulatory landscape and the fundamentals of anonymization, and then we'll have a period of Q&A. Now, thinking about why does it matter, data sharing. Some of you have know this very well, and for others of you, this may be new. But why does it matter that data being shared in the clinical trial space is being shared with the community in the numbers and scope that it is today? Research and science, when we think about it, is really continuing building upon itself and driving forward to the next discovery. In fact, Vivli, a global data sharing platform, really stands on the shoulders of those that come before us. Really, the knowledge of data sharing platforms, repositories, and leaders really depend on those who are generous with their time and experience. So really, big data-driven analyses offer a powerful approach for exploring complex diseases, such as Alzheimer's and other neurological diseases, for example. And someday we might be looking at diseases like COVID-19 and having those data shared in a platform like Vivli. And we'll talk about how that might work. We also look to reduce duplication of trials in a platform like Vivli and how that could work as well. And we'll talk to you about some examples here. And then to shape clinical trial design. So when I was running clinical trials for a CRO as well as a pharmaceutical company, it'd be great to have a platform like Vivli where that data, the raw data from clinical trials, was shared, and to be able to look at that data in a way to be able to shape clinical trials as they were being designed. And then perhaps most importantly, to leverage patient contributions. So patients are giving of themselves something very important, and then to be able to leverage that to answer more than just one question. More and more patients are expecting their data to be reused. And these quotes are coming from a meeting, a summit from Boston several years ago, and these quotes are directly from that summit. And we see that patients are really expecting their data to be shared after the clinical trials are over. The commitments are coming out of, some of you might be in industry, biotech, or pharmaceutical companies. And so those trade associations, whether it be bio, FPA, pharma, or IFPMA, have publicly stated commitments for their members. And then more and more journals and funders are requiring the raw data from clinical trials, the IPDs, to be shared more widely and not just with collaborators. So providing access to the individual participant level data really generates value. But to summarize, really honoring the commitments of participants, strengthening the trust in the clinical trial enterprise, preventing repetitive trials and putting additional patients at risk, and then enabling new discovery and scientific insights through combining data from disparate sources is how we move forward here at Vivli, and we enable that. But perhaps most importantly for participants, if that data is not shared, it is used really just one time to answer one question, most often the primary endpoint, rather than leveraging a participant's contributions to answer multiple scientific lines of inquiry, thereby advancing science. So when we think about balancing risks and benefits, we're really looking at balancing perhaps privacy, so protecting a participant's privacy versus more openness. So maximizing the value of the trial data that's collected to respect a participant's contributions. And that might be where you are right now. Should I share, or should I err on the side of more privacy? And that may be where we encounter most of the people that are trying to make a decision on whether to share, is how to balance these risks and benefits. So managed access data sharing platforms like Vivli offer a number of different things. What makes us unique is the combination of bringing a team together, an analysis space, and analytical tools, and a data repository. So managed access platforms protect data by bringing analytical tools and sensitive data together in what we call an isolated secure sandbox or research environment. Other terms for that are secure sandbox or cloud-based compute environment, rather than perhaps permitting download of the data for analysis. Coupled with that is typically a data repository, so a cloud-based compute environment. So that's not a replacement for deposition of data into a long-term archive for storage. So we use, our partner is Microsoft's Azure for long-term archive, and our platform is hosted in Microsoft's Azure cloud. So really, to introduce the entity Vivli, we are a nonprofit organization that offers convening for the industry, academia, and nonprofit funders and foundations. We offer harmonization around governance and policy and lower the barriers towards advocacy. And then we also have a user-friendly, secure, state-of-the-art data sharing and compute platform. So by the numbers today, we have over 4,900 trials that are available for sharing. That represents approximately 2.7 million participants from over 100 countries, and we represent 23 members that are part of Vivli, which means they offer their data available for sharing. So our secure environment bridges multiple platforms. This is an example of what that means. So a requester can sit within their office, or nowadays more at home, and can access data from multiple platforms. So this is representing, for example, several different pharmaceutical companies, and BioLink and Import are large government-funded NIH platforms, and really can access data from those platforms in a secure environment, and can access various statistical tools that we provide, like SAS, R, Stata, Python, and the Microsoft Office tools as well. So we offer 30+ software packages, and they can also bring in their own statistical tools as well. So we think about security as, pillars of security. So we have a data use agreement, the platform security that's built into Microsoft Azure, and then the third is, of course, data anonymization that will be that is provided by Privacy Analytics. Some of our members also do this on their own, of course. So data use agreement, some key provisions. So we manage the data use agreement process for members who use Vivli's harmonized data use agreement. The members who are partner platforms can also use their own data use agreement as long as it doesn't conflict with ours. in a process that we call cross-platform sharing. The key DUA provisions, the researcher agrees to, first of all, agree to a research plan that includes a scientific hypothesis, to make efforts to publish their research and not to re-identify participants. So our platform security is based on Microsoft's underlying security and then drive forward scientific research and data sharing. So in terms of a managed access platform, there are three major types: open access, that you can see on the left, managed access, and more restrictive access. The key features of an open access plan is really anyone can access these platforms through perhaps at times a simple account creation. Some of these do offer a simple online DUA or data use agreement. Managed access, which is what we are, really have an intermediary interposed and then require some type of scientific purposes that is required. So we require a proposal, independent review process, secure environment for data access, and a clear legal framework. And then restricted access is primarily invitation only and access only to those who provide the data. So that's how these three typically break out in terms of types. In terms of what data typically will be shared, these are the data packages that we have shared with us. So again, some top-line data, including study protocol, data dictionary, and statistical analysis plan, are typical, as well as the CSR, the clinical study report, and then the raw data itself, which is the final cleaned individual participant level data that is anonymized. And then there are some optional pieces that is also at times shared with us as part of the standard data package. I next would like to hand this over to Niamh McGuinness for sharing with her slides, and I'm really delighted to have her on board. So Niamh, I will pass that on to you. Thank you.
Thank you so much, Rebecca, and thank you for that wonderful presentation. I'm just going to ensure that I have the capacity here to make my own slides go through, which I don't currently seem to. Ah, perfect. I think I may have them now. Excellent. So hello, everyone. My name is Niamh McGuinness. I am a senior analyst on the clinical trial transparency team with Privacy Analytics. I'm delighted to be here today to be speaking with Rebecca. So I'm going to speak mostly today about the fundamentals of risk-based anonymization. So I'm going to start with defining anonymization and some of the situations in which it's required. I'm going to speak to the regulatory environment that's driving particularly anonymization of clinical trial documents, and how important it is for clinical trial sponsors to be kind of prepared to engage in these kinds of activities. I'll run through some of the differences between risk-based anonymization and rules-based anonymization, which are both currently actively used in this space. And I'm going to conclude with describing how we can use the 5 SAFEs framework to achieve effective risk-based anonymization. So anonymization can be defined as the removal or transformation of identifying information in a dataset such that data can be shared for secondary purposes without endangering the privacy of the data subject. Identifiers can be kind of broadly categorized as being either direct or indirect. Direct identifiers are not inherently analytically useful, and they can usually be used to directly identify an individual. So for example, name, phone number, etc. Indirect identifiers, conversely, are analytically useful. So things like demographic information, and as it pertains to clinical trials, medical history and treatment dates. So these aren't necessarily capable of singling out an individual alone, but in combination, there is a capacity for this to happen. Perfect. Okay, so particularly as it pertains to clinical trial document anonymization and public release, there are two main methods that we can use to handle patient identifiers. So if we consider this false sample narrative that may appear in a clinical study report, one way of protecting patient privacy is to effectively remove or redact all of the information. So in this way, we have managed to protect patient privacy, but so too have we removed all of the data utility. Instead, if we have decided to perform data transformation, we have this kind of capacity to maintain a degree of data utility. So here you can see that the subject identifier, instead of being removed, instead has been pseudonymized and replaced with a realistic looking replacement. This pseudonymized value could actually be applied throughout the documents and maintaining a degree of referential integrity. The medical history, instead of being removed, has been generalized to fall within the higher level group term in the MEDRA hierarchy. So there's still a degree of data utility there. And finally, the dates, instead of being removed, have been offset according to the Fuse algorithm. And this is particularly clinically useful because often the intervals between clinical dates are incredibly relevant. So these kinds of transformations can be applied according to a set of rules, or they can be applied in keeping with a statistical risk measurement. And when we use the outputs of statistical risk measurement to bring the risk of re-identification below a given threshold, this is called risk-based anonymization. So how do we go about assessing the risk of the risk of re-identification in a data set? So I'm going to use this kind of illustrative example to explain how we assess risk. So if we imagine that this is a breast cancer data set in the US, these three have been singled out because they're similar, for example, in their statistical properties. Maybe they have the same age, the same race, the same gender. So they're clustered together. Instead of having one Miss Orange, we now have three. And really, the larger the cluster here, the lower the risk of re-identification. So for example, if I were trying to assign the name Miss Orange to one of these three individuals, I have a 1 in 3 chance of correctly doing so. And this is an incredibly basic, but quite effective description of how we measure risk. So we can't really expect for a sponsor to have all of the data on a given population. It's usually just a sample. So when we look at this sample, we can see that there's only one person who looks like Miss Orange. But this doesn't mean that the risk for correctly re-identifying her is 100%. We actually have to look at the population as a whole. So we really need to use some statistical estimators to correctly estimate what the population is. In this way, we may be able to estimate that there are 2.7 people who look like Miss Orange in the data set, for example. Now, as you can probably imagine, this gets much more complicated depending on the type of data that you have. But irrespective of this, the same basic concepts will always apply. It ultimately all comes down to a clustering approach, gathering the numbers of people who are statistically similar in their properties. And despite the kind of apparent simplicity of this approach, it is incredibly well published upon. There are myriad guidance documents, academic papers, and books published on this topic. And after this presentation, if you want to reach out to me, I'd be more than happy to speak to you more about this. Now, there's more to measuring the risk of re-identification than just looking at the risk in the data. We also have to think about the context in which the data are shared. Just waiting for my slides to advance here. Rebecca, could you possibly advance the slide for me? I'm having a bit of difficulty here.
Did you see it advance?
I did not.
Okay, I'm sorry. It looks like it's advanced to me, but I'm not sure what happened.
Okay. Okay, I may just attempt. Apologies for this. Oh, it has advanced now. Okay, excellent. I may just have had a little bit of a perfect. Thank you so much for that. Apologies for that, guys. So as I mentioned before, there's more to it than just the inherent data risk. We can't really avoid thinking about the importance of the context in which the data are shared. So context is crucially important. And when we talk about context, we're really talking about the people that the data could be potentially shared with and under what circumstances. So it's obviously going to vary quite considerably depending upon whether the data are going to be shared in a secure enclave that has contractual and security controls in place, or whether the data are going to be shared on an open access portal, for example. So I'm going to speak a little bit about some of the regulatory circumstances in which anonymization is required. So I'm sure a lot of you are probably going to be quite familiar with some of these, but imagine we have a clinical trial sponsor who has both individual patient data and clinical study reports that may potentially need to be anonymized. Now, ordinarily, data sharing is performed under a voluntary basis. So with excellent initiatives such as Vivli, the data can be anonymized for sharing in order to advance clinical insights and to kind of contribute to the scientific community as a whole. On the other hand, I think it's pretty well established in the industry that anonymization of clinical study reports is quite onerous, and onerous enough really to dissuade people from performing it unless there is a regulatory requirement pushing them, such as EMA Policy 70 or Health Canada. And ordinarily, despite the fact that some sponsors will work on this themselves, sometimes they will opt to use a vendor like Privacy Analytics to perform these anonymizations. And so I spoke to the regulations that are pushing these kinds of activities. So Policy 70 was really a kind of a groundbreaking piece of policy, and it came into effect in January 2015. And what it called for was the proactive publication of anonymized versions of clinical study reports that were used in marketing authorization applications in the European Union. So what the policy said was there must be a demonstration that the risk of re-identification of the patients referred to in the documents must fall below a threshold of 0.09. Now, there have been successive iterations on this policy even since the EMA has had to move from London to Amsterdam because of Brexit. There have been continual updates to this. So Policy 70 activities are currently on hold, but I think there's an expectation that perhaps these things may restart this year or perhaps early next year. In that time of pausing of Policy 70 activities, Health Canada followed suit with an analogous policy in 2019. And it's very tightly aligned to Policy 70, largely speaking. It has the same threshold. But there's a really interesting addition to this, the addition for the potential of members of the public to request access to documents from older drug submissions. So this is quite similar to a provision made in EMA Policy 43, a separate policy. However, Health Canada differs in that these documents will not only be released to the requester, as with Policy 43, but also published alongside the proactive drug submission documents. And really interestingly, it's these on-request submissions for older clinical trial documents that are actually the majority of what is published on this portal, which I think is really cool because it kind of indicates that there's this public interest in clinical trials and what's happening in the clinical trial landscape. So I mentioned before that there are two main ways in which anonymization can be achieved, particularly as it pertains to Health Canada and EMA requests. So there's risk-based, which is what I'm speaking to here, and rules-based. So rules-based anonymization, really one of the downsides of it is that it can be kind of difficult to justify in case of regulator scrutiny because it isn't based on a quantitative measurement that can be compared to a threshold. It often results in the kind of the same standard anonymization that's applied to all data sets or documents, irrespective of the rare disease status and nuances like the context and variables. On the flip side, risk-based anonymization provides quantitative risk measurement that can provide a very strong assurance that it's comparative to a threshold and falls below that. You have a degree of control over data utility because you can adjust transformation of certain variables to give you more room to release other variables. And it is consistent with guidance from both EMA and Health Canada. And so there's actually explicit statements in both of their regulations that state that they would prefer a quantitative approach to risk measurement to be used wherever possible, as soon as sponsors are in a position to do so. And so that brings me really nicely to my next topic. What we've been seeing in the industry is sponsors really scrambling in order to meet these regulatory deadlines. Now, EMA timelines themselves are not very punishing right now because EMA are on sort of an extended hiatus. However, the Health Canada timelines, conversely, are very, very difficult to adhere to sometimes. You have only 60 days between the process initiation period and the production of the anonymized documents. So if you imagine you have a dossier that is, it could be 2 clinical study reports, or it could be 22, it could be 2,000 pages or 200,000. So if you have to try and mobilize an internal team, get the headcount, and find a workflow that has been proven and tested, that can be an incredibly challenging thing to meet in 60 days. Even if you were looking to outsource the work to a vendor, getting contracting up and running and getting the kickoff meetings set up and all of that work, 60 days is really, really a short time. So being prepared and being proactive about this is incredibly important. There's always the capacity for phase 1 of policy 70, which is the release of the clinical document phase of the policy. It is potentially going to resume in 2020 or early 2021. So I know that some sponsors probably breathed a sigh of relief because they had a bit of a break from performing these anonymizations. But we can't really rest on our laurels too much, because there's always a capacity for this to restart and for us to have to get working on this again. And then there's the kind of the often whispered about phase 2 of policy 70, which would necessitate and make necessary the sharing of individual patient level data in the same way for publication. So we have to be kind of proactive and considering all these potential areas for having to anonymize, because it could potentially be a pinch point for sponsors if preparation has not been made. So I've spoken about anonymization, what it's driven by, and how it can be realized as either redaction or transformation, and how it can happen in accordance with a rules-based or preferably a risk-based approach. So I'm going to conclude this part of the presentation before we move on to speaking about privacy analytics partnership with Vivli. We're going to talk about using the 5 safes, which is a fairly well-established framework. We're going to kind of repurpose it in such a way to demonstrate how you can effectively achieve risk-based anonymization. And so the application of the 5 safes in this way is actually is outlined in a white paper. It was written by our founder, Khalid, and our chief methodologist, Luke Arbuckle, and it will be circulated after the webinar. So let's start with safe projects. So how are we going to use the data? What are the ethical and legal requirements in using these data? The next, as I mentioned, is safe people. So we need to evaluate the level of trust that we have in the people who will be using the data. Next, we're going to look at the environment in which the data are going to be used. What is the environment in which they're operating? Are there security or contractual controls in place to protect the data and its use? And this is all going to contribute to how we evaluate the data itself. So not only are we going to look at clustering, but we also have to look at some of the complexities contained within the data and what we refer to as threat modeling, which I'll speak to a little bit later on. And this is all going to contribute to how we must transform or change the data in order to bring the risk of re-identification to an appropriately low level such that we can share or publish it. So let's start with safe projects again. So we need to understand where the data are coming from, what they will be used for, and under what circumstances. So what are the legal and ethical boundaries in place, etc. So this is going to set us up to decide the level of anonymization that we need to apply. So what is the appropriate threshold? And this is going to vary depending on the type of data and what it's used for. So the next thing is safe people. So who are going to be using the data? Who's going to access it? Will it be, secondary researchers in a secure enclave? Or will it be, in the case certainly of looking at Health Canada dossiers, anyone with an internet connection and say a passing interest in statins that have been licensed in Canada? So here we use the term adversary, which sounds aggressive, but it's actually an information security term, which we use to describe anyone who may either innocently or intentionally re-identify someone in a data set. So we need to determine what their motives and capacity might be to attempt to re-identify someone. And there are different types of adversaries. So one of those is an adversary that uses publicly available data to demonstrate that they have the capacity to re-identify someone. So an example of this was the HCUP case. So the HCUP was the Healthcare Cost Utilization Project. And it was a case in the US in which every state would provide data on all inpatient cases across a number of different hospitals. And these data were also longitudinal. So, there was information gathered at each kind of follow-up visit, etc. There were no terms of use, really. It was effectively public, and the data were very minimally transformed to protect privacy. And so what happened was that researchers were actually able to cross-reference media reports of vehicle accidents with these records, and it eventually led to the re-identification of 35 individuals. So that's an example of a deliberate re-identification attack, also called a demonstration attack. So it demonstrates that there's risk there. On the other hand, we have those kind of unintentional re-IDs that occur because a researcher or someone using the data might potentially know one of the data subjects. So if you think of all of the people that have what we call acquaintance level knowledge of you, it can be quite detailed. So it can range from relatives, spouses, all the way down to, if you're very chatty like me, the barista in your local coffee shop, you know, when we used to be allowed to go to coffee shops. And so even if this re-ID was accidental, you know, they still stand to learn something new about a re-identified data subject. So we really need to capture that scenario as well. The next thing we need to think about are the safe settings. So the environment in which these safe people are operating. So are the researchers using the data going to have special IT controls, passes to get into the building, to their office, etc.? Or, you know, will people have to fill out forms to state their intended use? There's many existing settings, or many existing standards, on how to assess risk in an environment like this, for example, like ISO, etc. And we can use this information to establish and assess the risk. So once we have the information on the people and the settings, we can establish what we call a risk matrix. So this looks really subjective, but there's actually expert probabilities that we can build into this, looking at previous data releases from reputable organizations. So we can look at individual cases, assess their context on the basis of these considerations. So really, when we're thinking about context risk, we're actually thinking about plausible re-identification attacks. So we have inadvertent attacks where someone with acquaintance level knowledge of another individual accidentally re-identified them. We have a deliberate attack, not dissimilar to the HCUP one that I mentioned earlier. And then finally, we have a breach. So a breach is kind of a catastrophic loss or theft of data. So to look at this holistically, again, we need to also consider the data. We also need to consider the context, and this will all contribute to the overall risk or identifiability. So great, we understand what the overall risk is. What's next? So we have to compare it to something to understand what an acceptable level of risk actually is. So how little risk do we need to comply with regulations? We can look at past precedents from data releases from reputable organizations and say that under these comparable circumstances, this was an appropriate level of risk. And this is going to depend on the sensitivity of the data, so how injurious it would be to have somebody be re-identified, or how much an adversary would stand to learn if the data were inappropriately used. So all of this is going to ultimately drive the degree of transformation that we will need to apply to data to meet these thresholds. So first, we know that we will need to transform direct identifiers, and certainly for anything like name or social security number, or for the Canadians, social insurance number, that has to go. But on the indirect identifier side, if information has analytic utility, we have to decide whether information could be publicly known or known more so to people with acquaintance level knowledge. And so we have to kind of change the data appropriately where necessary, depending on these inputs. And earlier I spoke to some ways in which data like these may be transformed, you know, for example, generalized or offset, etc. So the goal is to disassociate the data from the identities, and the other goal is to try and maintain a degree of utility in the data. So just to kind of conclude with this diagram once again, the whole purpose is really to evaluate the contextual sharing of data. So we need to know about the legal and ethical boundaries that are in place to protect the data. We have the individuals using the data, the safe people, the degree of trust we have in them, how safe the environment they're operating in is, all contributing to how we evaluate the data themselves, their statistical properties, the clustering, threat modeling, etc. And then from that, we can derive what an appropriate level of transformation is to apply to the data in order to ensure we have safe outputs that contain risk that falls below an acceptable threshold and make the data usable as well. So this risk-based approach that we use at Privacy Analytics, it's statistical, it's defensible, and it's evidence-based, and there are all sorts of publications that can be used to support this. So one of these is the white paper that this presentation is based on, which will be circulated, and there are many others like the High Trust De-Identification Framework. For example, again, please feel free to reach out to me if you need any additional references. So before I kind of go on to briefly talk about Privacy Analytics joint work with Vivli on a very timely topic, I just wanted to share my contact details. So please feel free to reach out to me if you're interested. My name, I know it's very odd, it rhymes with Steve, if that's helpful. So I'm more than happy to field any questions that you may have. So I've spoken a lot about risk-based anonymization. I'm just going to briefly mention the work that PA does. So we provide advisory services and data and document risk assessment and anonymization services. So we also have enterprise-grade software that we use in our own services and that our clients can use. So we handle all of these different inputs and produce outputs which have been anonymized in accordance with our risk-based approach. So obviously one of the very clear uses of these anonymized data is sharing the data, on a voluntary basis, as with Vivli, and this kind of work really helps contribute to research and transparency, and ultimately facilitates kind of clinical insights and innovation, which are great things. And in the spirit of facilitating clinical insights and kind of contributing to health research, at Privacy Analytics, we are going to waive fees to anonymize data from COVID-19 vaccine and pivotal drug trials. There will be a little bit more information about this in later slides. But for now, I would just like to thank you very, very much for your attention, and I'm going to hand you off to Rebecca again. Thank you.
Well, thanks so much, Niamh, for that extremely informative talk. It was really wonderful to hear the details behind your very important work at PA. And again, we'll take questions at the end. Coupled with what Privacy Analytics is doing, we are launching a COVID-19 portal. The objective of our portal will be to really lower the barriers. I'm sorry, I think I flipped into a different mode. Lower the barriers to data sharing during this time of public emergency. We have decided to waive fees to share, archive, and access COVID-19 trials that are shared within this portal. Together, what we've decided to do, and we welcome other partners as well that will be joining us, is to accelerate in some small part what we can do to accelerate the COVID-19 response through early data sharing of clinical trials. So again, I know Niamh talked about what they're doing in terms of the anonymization response, and certainly talk to them directly. I know Michael McTaggart and Niamh will be talking, and you can work with them directly, and inquiries can be made directly to Vivli or PA for this. So all members of Vivli will receive a discount on Privacy Analytics anonymization services, whether for COVID-19 or for other trials. We've worked together for the past several years and have standardized templates to work from, and we really very much value our partnership. Privacy Analytics can upload anonymization or anonymized data directly into the platform to help members reduce their workload. So at this time, we can certainly take questions from you. I know I certainly have a couple for Niamh, but I'll really defer to those of you. I can see there are well over 100 attendees, and I know there's probably some questions that have come through, and we can take those at this time. So if you have questions, please do use the question function, and we can take those at this time. So again, thanks, Niamh, for your talk, and I could certainly start us off and with some questions. I do have a question for you, Niamh. Is you mentioned that there are these 5 safes, and that was really interesting, and I have read about this already. The level 5 risk. You talked about this, and one thing you mentioned is the risk level. You talked about having to be below a certain appropriate level, but you didn't talk more specifically about, what that level might be, and maybe you can talk about what is that level of risk that you are most comfortable with, since you do have a quantitative risk level that you
Sure.
mentioned?
Of course. Oh, thanks so much for that question. So I think the threshold that we work under most frequently, and this is really belying the fact that I work very much on the document anonymization space. It's 0.09 is the industry mandated threshold for what's an acceptable degree of identifiability. But of course, it varies massively depending. That's for a public release of clinical trial document information. So it varies massively depending on the context of release and depending on the sensitivity of the data. So, an example of this is when we're working with a sponsor who is preparing, say, data for anonymization for upload to, a portal like Vivli, etc. We have to do this pretty strong context assessment. So it's filling out a number of forms to evaluate all of these different pieces. And this is going to be used in tandem with us looking at the data themselves and how sensitive they are. And these are all going to determine what the appropriate threshold is. So, for regulatory cases, we can't really move off of that industry mandated threshold of 0.09. We need to stay below that. But certainly in more nuanced cases, we can kind of decide on the basis of past precedents, the data themselves, and the context in which they will be released to determine what the threshold is. I actually have a question for you as well, if that's okay, and please feel free to ask more in the meantime. I was thinking about this, recently. I think we're probably all thinking about this constantly. I think Vivli is in a very unique position to contribute so massively to clinical insights and sharing of data, and allowing a kind of a consortium of people to get together and share insights and potentially develop ways of treating this horrific pandemic. Do you think that the situation we're in is going to highlight for those pharma companies that really haven't been that proactive in establishing data sharing efforts? Do you think this is going to highlight for them how important it is to be doing this in a kind of a more proactive manner? Or do you think that in the event that these things, the vaccines get developed and a treatment is established, do you think things are going to return to normal? Or do you think this is going to have kind of an indelible effect on the industry as a whole?
Well, I actually think it will be very hard for the entire world to go back to normal after this, right? To business as usual. And I think as we've worked with, various members, each one of them have what I call their own risk personality, just like each individual has their own tolerance for risk. And I'm not sure that the COVID, crisis will change that. Some of them may change their risk profile based on that, and some will, maybe rise to the challenge and realize that they do need to share more, and some of them will be more risk intolerant. We've certainly had a increase in the number of inquiries since this has come up, and I feel like that people are maybe less competitive and realize that they do need to share more as we're combating, a common really challenge. So I think that's certainly a good thing. That's a really great question, Eve, and thanks so much. So we do have a question that's coming up in terms of re-identification risk, and I think that you should probably be the best person to take that, is how is that applied to genetic data? If you have a small number of rare SNPs that are unique for an individual but are in no way observable, how would that carry a re-identification risk? So I think that's an interesting question, right? Because rare
a really interesting question.
Are ones that we are getting inquiries about all the time, and that is an interesting question.
Absolutely, and thank you for this question. This is actually quite topical because I had a meeting about this very recently. We're trying to establish a kind of a best practice for dealing with this kind of information, because we have now encountered it both in our document anonymization and in our data anonymization. It's continuing to come up kind of more frequently now. So there's two elements to this. On one hand, for something to be deemed identifying, it has to be knowable. So I don't even know what my blood type is, which is terrible. I've completely forgotten what it is. So I certainly probably wouldn't have, and I have an immunology background. I certainly wouldn't know what my particular genetic predisposition is to certain things. That being said, SNPs can be incredibly unique to an individual, often can be unique to an individual, almost an entire population. So we have to consider that as well in terms of how much it can distinguish one person from another. So right now, what we would do in order to be conservative would be probably to transform that information in the most stringent way possible and thus remove it. But I agree that going forward, that probably isn't the best way of handling genetic data for the means of analysis. So we're actually trying to establish a framework right now on how best to handle these data as it stands right now. So I think the best thing here is to stand by, and you'll probably see publications from us coming out about this particular topic in the coming months, because it's actually been something we've been talking about quite a bit. But it's a really interesting question, and thanks for that. Another question that we've been asked a lot is how we handle images, so MRI data and data from imaging, you can't necessarily re-identify someone on the basis of their, what their brain looks like. But we are kind of finding a way to handle with, particularly the identifying information on these DICOM images and how best to de-identify these and keep the clinical relevance too. So this is very much the bleeding edge of risk-based anonymization, and certainly stay tuned, because we are going to be publishing and speaking about this more in the coming years.
Absolutely. Okay, wonderful. So I'll take the next one, which is, how do we handle sharing of data in ICF, informed consent form, particularly for older studies that might not have contemplated such sharing? So that's an excellent question. That's one of the most common questions that we get is do people review the informed consent forms? Clearly, most of the data in Vivli, which we have almost 5,000 trials, these trials, there's really excellent broad consent form language for data for trials that are started today or maybe a year ago, but the trials that have hit us now, we really did not contemplate data sharing if they were started 5 or 6 years ago. And how did we manage the informed consent language, right? And best practice really thinks about how patients, and there are many papers that we can circulate after that, that did not contemplate such sharing. Our answer is really that best practice really says that data can be shared unless the informed consent form said that data could not be shared. Many of our members have, based on their risk tolerance, do not look at each and every informed consent form, but a minority of them do review the informed consent form to see if that data said that it could not be shared. So in a small percentage of the informed consent forms, those forms say that data could not be shared. For example, it might say that the genetic data from this trial could not be shared, or the vast majority of the informed consent forms are silent. So many of our members do not review the informed consent form. So again, it's really the risk tolerance of our members of whether they review each and every informed consent form from each and every country. So there is no kind of uniformity on whether the informed consent forms are reviewed. That really varies by member. So I'm going to ask Neve another question that has come through, and using rule-based anonymization, can you differentiate between which rules to apply depending on whether the trial is a rare disease or not?
Yeah, so this was a question, well, it was more a comment that I was probably a little bit unkind to rules-based anonymization, and there is the capacity to actually apply different rules depending on rare disease status. My comment was really, it wasn't, attacking a rules-based approach in any way. It was just saying that it is more difficult given that there isn't as much flexibility in achieving data utility as there is with a risk-based approach, because you don't have a quantitative measurement that you can look to and adjust. It was just more a comment that in the absence of using a quantitative approach, it is more difficult to take into account these nuances. Not impossible, but certainly more difficult. So thank you for that comment and for keeping me in check there.
Absolutely. And then maybe we can take this one together, but the question is, can the de-identification process help researchers conform with the GDPR guidelines in Europe? I mean, we did have an entire webinar devoted to GDPR and anonymization with our, so there's, really the legal aspect of how anonymization really helps, and consent helps us with GDPR, but maybe you have a different answer to that, Neve, and I'll let you take that as well.
No, I think your answer was great. We have written a lot and spoken a lot about the application of GDPR in order to achieve this kind of compliant anonymization. We actually provide advisory services on this, so this is something that can be applied in order to conform with GDPR. And in fact, I think Khaled wrote a paper in tandem with a legal team to speak to this specifically. So absolutely, data anonymization can be applied in order to conform with GDPR, and in fact it can be applied to conform with all sorts of regulations. We just have to apply a different set of criteria to our de-identification to make sure that we conform with that.
Okay, wonderful. And then there's a couple questions regarding the COVID-19 and how to access data from patients for COVID. And, we are launching this COVID-19 portal. So, if you check back, you should be able to access data from that. And the question is that would be including the clinical trial data. Those data would be, the trials have not yet, some of them have not started yet, and obviously that data would be forthcoming. So that's really the answer to that. One question for you, Neve, is for repositories with very large collections, are there automated or sophisticated ways to implement risk-based anonymization?
The short answer to that is yes. So our tools have now been built in order to kind of best handle automated classification of variables in standardized kind of CDISC compliant format. So, if the data have been kind of cleaned and collected in such a way and formatted such that they kind of comply with modern data collection standards, then absolutely. So there's actually right now there's a discussion happening with the Fuse group about how to best practices for data collection and gathering in order to kind of expedite anonymization and how to handle that. So that's a very topical thing. But yes, I think it certainly can be automated. We just have to have an understanding that the data have to be in a certain form before we can do that. And that kind of applies to the work that we will do on pivotal or vaccine trials for COVID. We're going to waive fees, but there are conditions in place in order to make it operationally efficient and fast and to try and have the data be anonymized as quickly as possible. We do need the data to be in a certain form before we can achieve that. But certainly I think it's achievable.
Terrific. And currently the one more that's come through is the European Open Science Cloud. Many of us have heard about this. It's a very ambitious effort. The EOSC is built up, and there's really a strong need for tools and services for handling the very sensitive data, such as anonymization. What could privacy analytics do for this effort?
Well, we have enterprise-grade software that deals with anonymization of large clinical data sets. So we could potentially contribute to that, absolutely. So I'd be really excited to hear more about that. But yeah, we could. That's something we could certainly do. And we have consulted with clients on setting up kind of overall projects and large programs in order to achieve kind of high throughput, high efficiency, scalable and automated anonymization. So this would be something that we could potentially do as well. Great question.
Great. Okay, and then there's a question coming in about data proxies. So those data points that seem innocent, but then actually could reveal sensitive inferences using analytical methods such as, machine learning. Any thoughts on that? Because, that's really kind of the cutting edge of what we're seeing and has been written on quite a bit.
I know that these questions are fantastic. They're really great, and I can't wait to speak to them more with our broader privacy analytics team. When we receive a large data set and it contains things that we haven't seen before, usually we have kind of a methodological discussion, and we talk about means of best handling this in the future. So machine learning and AI are definitely integrated into both into our software and into our services offerings. So this is something we're actually looking into as well, to try and handle the things that seem innocent but are potentially very identifying. So that is something that's on our radar, absolutely.
Terrific. And then, what about the risk threshold? You did talk about that at 0.9, but could you explain a little bit about how the factors are really identified and given a value based off of that?
Sure. So the threshold of 0.09, I mentioned before that in many cases, these thresholds are set on the basis of past precedents. So, and that's really the main way in which they are determined. So we look at past data releases from reputable sources, and we look at the use of the data and how it was consumed, and the capacity for re-identification, and determine whether or not the threshold used then was appropriate to kind of propagate throughout. So just to kind of bring it down to basic principles, using a threshold of 0.09, it means that a person has to look like 10 other people, so an equivalence class of 11. Now that is very simplified, and I know there's probably some biased statisticians that are very angry at me for saying that. So that is the level of identifiability that is thought to be acceptable for a public release of clinical study reports. So you have to remember too that this could be changed depending on the sensitivity of the data. So 0.09 is a general rule, and it is what EMA and Health Canada really want us to stick with. But imagine a case in which you have a data set, and this is and I'm sorry to bring this up, but this is not a nice thing to talk about in the morning. But say a data set about HIV transmission in children or something like that. That's an incredibly sensitive collection of data. And so in some cases, we need to enforce a more stringent threshold based on that. And that usually will come with kind of large discussions and determining what the appropriate course of action is. So the thresholds are subject to change. I think EMA and Health Canada have been implementing this 0.09 as a method of kind of keeping things simple. And certainly there are cases in which the threshold should be increased or decreased. So it depends on past precedents. It depends on the potential situation. There are a lot of inputs to it, the data themselves, their sensitivity, and also the context of release. So it always varies. Depending on the unique situation, but certainly it's not always appropriate to use a threshold of 0.09.
Absolutely. Okay, so that makes sense to me, and thank you so much. Well, here's my thought.
Thank you.
We do have a few more questions, but I do think that we should respect everyone's time. And if you have additional questions, please send them through directly to myself, Julie, or I know Neve gave her contact information. We will certainly send any questions on to Neve. And really nicely done. Appreciate the thoughtful responses, and thank you all for being wonderful participants. I know it's a challenging time for all of us. I wish you health, and thank you for your participation today, wherever you are in the world. And a slide and a link to today's recording will be circulated via email. Please do feel free to distribute them. We do hope you'll join us for the future webinars. And thank you for joining today, Neve, and all of you. So again, thank you, and wish you all well.
Thank you all so much.
Thank you. Take care. Bye-bye.